Hello guys, today we discuss about the history of microbiology. So what is microbiology? Microbiology is the study of microorganisms, microbes which is visible only on with the microscope. The diverse group of organisms include the algae, archaea, bacteria, cyanobacteria, fungi, protozoa, viruses. Most of the microorganisms are harmless. 99% are the good, that is example is the cyanobacteria, that is the blue-green algae, and 1% are the bad, that is the pathogens. And pathogens are responsible for the every disease. So microbiology, in which we study discovery era, transition era, golden era, and the modern era. First one is the discovery era, spontaneous generation. So in which was first one is the Aristotle scientist, and other believe that living microorganism could develop from the non-living materials. In 13th century, Rogan described that this is caused by the minute seed or the germ. Anton van Leeuwenhoek, in its time span is the 1632 to the 1723. Description of protozoa, basic types of bacteria, yeast and algae. Father of the bacteriology and the protozoology. In 1676, he observed and described microorganisms such as bacteria and the protozoa as the animalcules. The term microbes is used by the Sedlot in 1878. Next one is the transition era, in which the Francisco Redi, his lifespan is a 1626 into to 1697. He showed that maggots would not arise from the decaying mat when it is covered. Next one is the John Needham, supported of the spontaneous generation theory. He proposed that tiny organisms, that is animalcules, arise spontaneously on the mutant gravy. He covered the flask with the cork as done by the Redi. Still the microbes appear on the mutant broth. Next one is the Lazzaro Spallanzani. He demonstrated that air carried germs to the culture medium. He showed that boiled broth would not give rise to microscopic forms of the life. Next one is the golden era, in which Louis Pasteur is the scientist. He is the father of the medical microbiology. He pointed that no growth took place in the swan neck shaped tube because dust and the germs had been trapped on the walls of the curved neck. But if the neck were broken off so that dust fell directly down into the flask, microbial growth commenced immediately. Pasteur in 1897 suggested that mild heating at the 62.8 degrees Celsius, that is 145 degree Fahrenheit, for 30 minutes rather than the boiling was enough to destroy the undesirable organisms without running in the taste of the product. The process was called as the pasteurization. He also invented the process of the pasteurization, fermentation, and the development of the effective vaccine, that is the rabies and the anthrax. Pasteur demonstrated disease of the silkworm was due to the protozoan parasite. Next one is the contribution of the Louis Pasteur in the microbiology. He coined the term microbiology, aerobic and anaerobic. He discovered the theory of spontaneous generation. He demonstrated that anthrax was caused by the bacteria and also produced the vaccine of the disease. He developed live attenuated vaccine for the disease. Next one is the John Tyndall. He discovered highly resistant bacterial structure later on the endospore. Prolonged boiling of the intermediate heating was necessary to kill these spores. Next one is the Lord Joseph Lister. He is the father of the antiseptic surgery. Lister concluded that wood infection too were due to microorganisms. He also devised a method of the destroy microorganisms in the operation theater by spraying the fine mist caprylic acid into the air. Next one is the cost postulates. There are the four postulates. First one is the same microorganisms are present in the every case of the disease. Second postulate is the microorganisms are isolated from tissue of a dead animal and the pure culture of the is prepared. Next one is the microorganism from the pure cultures are inoculated into the healthy susceptible animal. The disease is reproduced. And the last one is the identical microorganisms are isolated and the recultivated from the tissue specimens of the experimental animal. So these are the four Koch postulates. Next is the Fem Ishmael Hayes. One Koch assistant first proposed the use of agar in the culture media. It was not attacked by the most bacteria. Agar is better than the gelatin because of its higher melting point, 96 degrees Celsius, and the solidifying is the 40 to 45 degree Celsius points. Richard Petrie, he developed the Petri dish, that is a plate, a container used for the solid culture media. Next one is the Edward Jenner, first to prevent the smallpox. He discovered the technique of the vaccination. Alexander Fleming is the discovered the penicillin from the Penicillium notatum that destroyed the several pathogenic bacteria. And last one is the Paul Ehrlich. He discovered the treatment of syphilis by using the arsenic. He studied toxins and the antitoxins in the quantitative terms and the laid foundation of the biological standardization. So, to teach is to learn. Thank you.